Okay, sure. Okay, thank you, and this is the last session. So when Maxine and Meredith invited me to participate here, my understanding was this was a reset of biodemography. It's a bit like what Richard Sussman talked about. And so that's the spirit in which Brian and I, a young colleague in my department, and I put this paper together. So what I'm going to speak about is the social insects, rather than the fruit fly. I specialize in fruit flies, so that I'm talking in an area that I don't have great depth of knowledge here. And it's going to be essentially, in some ways, like a tutorial on social insects. My overall goal is to give you a sense of possibilities of the research using social insects in this sort of new direction of biodemography. And I've organized this into 4 different parts. First, I'll give an overview of just social insects and longevity and so forth. And then second, the evolution of sociality in wasps. And then thirdly, a bit about the honeybees, overview of honeybee biology, and then lastly, some research areas that I see as possibilities here. And I see also, just to set the stage too for possibilities, huge possibilities, because here I'll give you a sense of the diversity of species, a gradation from the pre-social to the eusocial and so forth. So there's huge possibilities for research in comparative biology and demography and biodemography of social insects. But also there's a deep pool of young talent. I mean, Olav Ruppel right here, Brian Johnson, for example, and some of the best and the brightest go into research on social insects, and they're passionate about their work. So there's an untapped pool there that I see as great possibilities. So, and there's Brian Johnson, and new faculty in our department. He studied under Tom Seeley at Cornell. So anyway, here's the social insects. They fall into 4 categories. One, the ants, wasps, bees, and termites. So I have pictures there at the bottom. There are 2% of the 2 million insect species, but it's basically 80% of all insect biomass, and they outweigh the vertebrates by 7-fold. I mean, social insects, I mean, ants, you could run outside here, and I mean, they're, let's see, what is this, April? Anyway, they're out there. And there's a lot of social insects, we all know that. And basically it's sophisticated problem solving resembles voting. And so there's just really interesting questions here. How you have just individuals that end up building these termite mounds and really sophisticated nests and so forth for bees and ants. And they're adaptively allocated to different tasks. The processes are regulated by interactions, and they're excellent models since you can individually track these, so that you can't get into ants species where you have tens of thousands of species necessarily, although you can do that some. But you can get some of these species have fairly small numbers, and you can bring them into the laboratory and really study these at the individual level. Okay, so sociality, just for a little background here. This is basically, there's 4 different criteria here. Continued care of young. You have cooperative brood care, so it's not just a single individual, a queen or a mother. A reproductive division of labor, and you need colonies with at least 2 adult generations. Okay. Now the different levels of sociality, you have subsocial, you actually even have pre-social. Whoops, here. Back up. Okay, I guess I didn't. Okay, subsocial, you have colonial, you have communal, you have cooperative breeding, and then you have eusocial. All right. So here's a paper by Keller and a colleague came out in '97, and give you a perspective on the longevity of social insects relative to solitary insects. And so this is a mean lifespan of 87 solitary insect species. From 8 orders and 61 species of eusocial insects. So you see lifespan from 0 to 30 in years at the bottom, and there's a number of species there at the top. And so there it is. I mean, this is actually not quite right in my view. At least, maybe, I guess it would be for those 87 they picked. But some of these, non-social species can live beyond a year. But in any case, they're not going to live 25 or 30 years. And then you can see here with these social species, eusocial species, whatever, it's 1 and 2 years up to 30 years or so, you get into ant queens that live incredibly long time. So there's an overview I put together a number of years ago. It's more schematic than data per se, but it's insect lifespans. And so it goes from the most primitive on the left to the most advanced, at least for the insects, the Hymenoptera on the left there. But anyway, you can see there's the Isoptera, and so this is the termites, and so that you have some of the worker termites living weeks to a few months, but then you can get the queen termites that live in these huge mounds deep in the below the surface, subterranean, that live a couple of decades or more. And then you have the Hymenoptera, which are the ants, bees, and wasps. And the parasitoid wasps, some of these just live a few weeks. This is I'm going to actually walk through the evolution from that most primitive wasp all the way through ant queens that live, again, 20 or 30 years. So you note here that, well, I guess I have another schematic here for the different categories looked at alone. But anyway, the wasps, here you are, months and years. You have the bees, again, months and years. But then you get the ants, where it goes from weeks to decades. Same thing with termites, from weeks to decades for the queens. So you see there on the right, with the ones that live decades, are the subterranean ones. Okay, you don't find 10 and 20-year bees and wasps. Okay, and there at the bottom you can see sort of a range of workers in terms of longevity, bit of an overlap and there. But anyway, clearly the queens are the really long-lived ones. Okay. And so just a summary here, you have lifespans within this group that differ by 800-fold. You have differences both within and between these different groups. Males are typically short-lived, and the lifespan and colony size are uncorrelated. Okay, I guess I have arrows there, sorry. Okay, so look, here's, there's just a classic paper that I'll summarize here, next slide or two, by famous entomologist Howard Evans back in the 50s on the evolution of sociality in wasps. And, but anyway, so what I do is set the stage here with basically the wasp floor plan. How do you get from here to there kind of thing in the wasp? So look, wasps, I mean the most primitive species or group of species in this in the Hymenoptera are the sawflies. They're not wasps, but they're precursors to the wasps. And basically they have a precision, you can see it laying an egg in a stem there. They start out with precision oviposition. That's the start of the sting. Okay? Then you get the parasitoid with the sting. All right? Now I'm going to talk about wasp waste in just a couple or three bullets here. But, you know, this is so they can manipulate their abdomen and, you know, sting these caterpillars or spiders or whatever they're going after. That's where this wasp waste concept comes from. This is important in the bigger evolutionary picture. You have mass provisioning concept, and that is where the wasps sting a prey, in this case a caterpillar, drag it back to a hole, and lay an egg on it. That's as distinct from progressive provisioning like birds. They get to that later on in the evolutionary sequence. And then mandibular mouthparts, you don't think, in entomology you think of sucking insects and chewing mouthparts and so forth. But in fact, this is another important concept because they can use their mandibular or chewing mouthparts that were part of the sawfly and so forth to build, you know, pretty sophisticated nests. Then this wasp waste basically restricts the nourishment to liquid. You can't get very much through that little tiny waste, and so it's liquid. And this really pre-adapts them for social evolution because it's liquid, it's exchange of liquids in order to communicate, okay? So the liquid exchange for within colony evolution, basically. So there it is. That floor plan set the stage for the evolution of eusociality in wasps, okay? Now, Howard Evans had 13 different stages, and I don't cover all of those here, but just to give you a flavor for sort of the progression here, you start out with these parasitoids that sting a caterpillar or whatever, and they lay an egg on it, or I mean, sorry, in it, and so it becomes a living incubator. But unlike that wasp I showed you with the caterpillar before, the earliest ones, they simply sting it and then they fly away and find another and sting it. And so that you have a lot of mortality because the caterpillar, that is the incubator, is still out there to be, you know, preyed upon, and the wasp is out there sort of hanging out too, no nest or anything. But now the next stage here would be just a really primitive nest, so that the wasp would, again, sting a prey, drag it back maybe to a really primitive nest, and again, mass provision, just lay the egg on it. But now the mortality conditions change because it's not exposed, and the mother or the female can hide there and hang out at night. The same thing here. This would be the next stage where, again, they're mass provisioning, but it's a more sophisticated nest, and you know, lay their egg on the host and seal it, and so forth. And then this can get into where you have a single port of entry. And where do we go the next? Okay, here's the next one where you have the egg, lay the egg before you have any food. So this would be like a paper wasp where you lay the egg in the cell, and then you go out and get some prey, masticate, and then you regurgitate it or whatever to the developing larvae. So now you're into progressive provisioning. It's like bird feeding. Here would be another kind of variation on this, although it's the next stage where you have a single entry. And then finally, the final stage, when you get into eusociality, where this would be like a yellow jacket or whatnot, where it's really sophisticated nests, and again, the progressive provisioning, divisional labor, and so forth. So that, anyway, that's a progression. So now, here would be a sequence, again, not the sort of, you know, grouped into broad categories here. The first one is the emergence of the nest as a locus. So now this is, you know, central, what they call central place foraging, where they all come back to the same place. This is really important for sociality. And so anyway, the nest concept's important. Then the emergence of parental care, that's again like the bird feeding with progressive feeding. And then lastly, the emergence of the queen concept. This emerges before this last stage, of course. But anyway, there's a stage in here when I was reading this early literature where Evans talked about the life of the female is a prolonged overlap with the offspring. And of course, as a you know, person who does aging research, this caught my attention. And really, this is the threshold of sociality, because you have to have a mother alive at the same time her daughters are emerging. Where you have the mass provisioning in earlier stages, the mother, you know, lays the egg, seals the hole, and flies away, and probably dies before the offspring emerge. But here, you have the mother alive with the offspring. Now you have incipient sociality, and now you're off to the races, you know. So anyway, one of the carry-away messages here is that longevity was a precondition, you know, extent to longevity, a precondition for the evolution of sociality. So now that set the stage. So what you have is extended longevity is a precondition for the evolution of incipient sociality. But sociality, in turn, creates conditions for the evolution of further extension and longevity. So now you have a division of labor, you have a nest, you can you know, have some that guard the nest and others that go forage and so forth. And so that you just get a you get a feedback loop where, you know, greater longevity sets a stage for greater sociality, in turn sets a stage for greater longevity and so forth. That's how it works. Okay. Anyway, this is another concept that I'll just throw out. It's one of the few original things that I actually present that I, you know, sort of contributed here in the historically anyway. But when I was teaching a course in insect demography way back when I first started, I asked the question, what's different about social insect demography than just regular, you know, solitary, whatever. And of course, that's the basic linear model here is birth minus death, you know, times the number of the population equal to growth. And so they said most of the bee people just simply, I don't know, maybe this, I think this is true. Anyway, they, you know, just said call your individual the colony. And I just, well, that's not really very interesting. So, but anyway, the simplest model here is where you have the number of workers, dn dt equal the birth rate of the queen minus the death rate of the workers times the number of workers. Okay, so what you know, the concept here is that the colony can't grow until the female first replaces those that die. So if it's a small colony, then most of what she produces goes into growth, and there's a few to be replaced. But as a colony grows, you have big numbers, the same per capita death rate, but in fact you get into big numbers where all you're doing is just replacing those that die. And so you can no longer grow unless you swarm. And so what's that maximum size? It'd be simply set that to 0, and you have the birth rate of the queen over the death rate of the workers equal that maximum size. So you have a demographic limit as distinct from a tree hole limit or something, which is that. But also the inverse of death rate is expectation of life. So it turns into this simple, why didn't I think of this? Because it's so simple. The number born times the expectation of life gives you the number of, person years or bee years in this case. So anyway, there's a maximum size to a colony. And by the way, then Tolja used his mathematical magic to take this to all new levels where you have now age structure of individuals within colony, but then the age structure of colonies is really, we published this, one of the students took the lead anyway in ecology about 3-4 years ago. Okay, so there's a number just for perspective that this actually works. That the queen bee lays about 1,000 eggs a day, and you have a 50-day worker lifespan, and 50,000 sort of at the upper size of a honeybee colony, but at least it's ballpark. This isn't just theory. Okay, so biodemographic principles, you have generation overlap as a prerequisite for the evolution of incipient sociality. This progressive provisioning evolved after the evolution of extended longevity. Sociality provides the insurance-based survival advantages, so you have division of labor and among other things in the nest and that kind of infrastructure, which increases survival. And then this queen worker longevity differentials. This does not work so well. And so when the colony's small, that every worker retains its ability their female workers, to reproduce, because if the queen dies, they have some chance to replace her. But when you become, 20, 30,000, it's an absolute lottery. And therefore, at that point, or somewhere along the line, you find that the workers lose their ability to reproduce, and also they're shorter lived, and the queen becomes the long-lived individual. It's because you've gone from different levels of individualism. That is, you grade into the superorganism as the individual rather than just the parts. And so the queen basically becomes the ovary of that superorganism. So anyway, there's a picture we can include in our paper there of the marked ants, and really interesting. You can see how they have marked pebbles on the right there and so forth. But anyway, okay, honeybee organization and communication. So there's we have honeybee colony organization, castes, and there we go, communicating, and then communication to make honey. Okay, now organization of the bee colony. You have 4 basic age castes. You have the newly emerged, you have the nurses, you have the middle-aged bees, and you have the foragers. So this is a little tutorial on honeybee biology here. So what is a temporal caste? You have different ages, specialized at different castes. The physiology strongly changes to specialize the bees for their castes, and the bees go through puberty. I don't know if that's used much in bee biology, but anyway, that's what Brian used. Anyway, but you can see at the top they're foraging, and at the bottom they're nurse bees, basically. Yeah, I guess I have a pointer there. And the different castes have different physiologies, so that these hyperpharyngeal gland side, this is basically a protein synthesizing gland, and versus the age. So you see the newly emerged, very small, the nurse, which is synthesizing protein to give the larvae to grow and so forth, and then middle age, these are the ones that are producing honey basically, and then the forager, they don't have to worry about that, they're out foraging. And so one of the things you can do with bees is that you can manipulate the colony so that they all become nurses, so the middle aged become nurses, so you can reverse aging and these sorts of things. It's a great opportunities for manipulating things like that, where you can make them younger and back into nurse cells, so you remove all the newly emerged ones, or all the nurse ones, and then the middle age have to go back to where somebody has to mind the store and the babies and all that business. Okay. Now, newly emerged bees clean the cells. They get the dirty work, like a lot of the young people, right? Okay. Anyway, they clean the cells, continue to develop. Then you have nurses. They feed the queen and the young. And there's a developing larvae, by the way, so you can see this. This is progressive provisioning, just for perspective, if you haven't seen a beehive like this, but those are the maggots, basically, or the larvae right there, and the bees are tending them. Then middle-aged bees are basically processing food, building the nest, and guarding the nest. And then the foragers are, of course, going out and getting nectar and pollen, okay? And how do the casts all work together? That's a big, these are deep questions on this material right here. Is that there's a big part of the paper that Brian put together, that's more certainly a specialty here, is where you have social insects as models. And I just talked about this earlier. You have wide variation in lifespan, you have large variation within the species, you can decouple aging and reproduction. Most species, I mean, solitary, you have a cost of reproduction, but in fact, in social insects, you have, there's apparently not a cost. I mean, queen bees somehow gotten around that, and they can lay eggs their entire life. And also one other point here was this high expression of antioxidants is shown to be unnecessary for a long lifespan. Okay? I mentioned this just briefly before, how you can regulate aging through manipulation of the colony. There's several, let's see if I have arrows there, I don't. Several physiological states so that you can use those to study. Nurse to food stores to foragers. Nurse to food stores to foragers. Anyway, you can manipulate that so that you can change their role, but also their aging rate. You can have bees kept in the nurse phase for several months, whereas foragers live only 2 to 3 weeks. There's evidence that the foragers turn off their repair pathways, and there's winter bees that live 6 months, and the physiology of these bees is similar to the nurses. Here's the research opportunities as we put together here. This is more or less my last slide here. And that is, one, you have integration of colony, organismal, genetic, and molecular level on health and aging longevity. So you have at the societal level, that is the superorganism level, right down to the genetic and molecular level. Huge possibilities there for understanding when you tweak mechanisms, what happens at the next several levels. Big possibilities there. This flows from Ron Lee's work, intergenerational transfer, health and longevity. There's certainly possibilities there. Interdisciplinary research at the individual level. It's just fascinating that tracking these individual ants in this case, but the bees or whatever, that the information you get when you have it at the individual level. And then, of course, a comparative biodemography of social insects, as I walked through from the pre-social all the way to the eusocial. And another one here is the honeybee health. This colony collapse, everybody's heard about this. I call it like a ghost ship. One day they seem to be all there, and you go back and say, where did they go? And they still don't have a good handle on what's, it's probably multiple factors, it's kind of what they think it is, but this colony collapse. But anyway, there's health people working in honeybees that are concerned about health, just like we're concerned about human health, that would map into sort of the next generation of biodemography. I think that's it. Okay, thanks very much. Second, the evolution of sociality in wasps. And then thirdly, a bit about the honeybees, overview of honeybee biology, and then lastly, some research areas that I see as possibilities here. And I see also, just to set the stage too for possibilities, huge possibilities, because here I'll give you a sense of the diversity of species, a gradation from the pre-social to the eusocial. Okay, sure. Okay, thank you, and it's the last session. So when Maxine and Meredith invited me to participate here, my understanding was this was a reset of biodemography. It's a bit like what Richard Sussman talked about. And so that's the spirit in which Brian and I, a young colleague in my department, and I put this paper together. So what I'm going to speak about is the social insects. And there's Brian Johnson, and new faculty in our department. He studied under Tom Seeley at Cornell. So anyway, here's the social insects. They fall into 4 categories. One, the ants, wasps, bees, and termites. So I have pictures there at the bottom. There are 2% of the 2 million insect species, but it's basically 80% of all insect biomass and so forth. So there's huge possibilities for research in comparative biology and demography and biodemography of social insects. But also there's a deep pool young talent. I mean, Olav Ruppel right here, Brian Johnson, for example, and some of the best and the brightest go into research on social insects, and they're passionate about their work. So there's an untapped pool there that I see as great possibilities. So than the fruit fly, I specialize in fruit flies, so that I'm talking in an area that I don't have great depth of knowledge here. And it's going to be essentially, in some ways, like a tutorial on social insects. My overall goal is to give you a sense of possibilities of the research using social insects in this sort of new direction of biodemography. And I've organized this into 4 different parts. First, I'll give an overview of just social insects and longevity and so forth. And then